Hello, good morning, and welcome to all of you. This is Rajesh Kumar Singhal, and I am here to present a new lecture on the phytochelatins. So, friends, phytochelatins means these are the chelating compounds. These are the chelating peptides which have important role in the chelation of metals, mainly heavy metals. Okay. So what is the heavy metals? So these are the elements which have density more than 5 gram centimeter cube. Okay. And there are some D block compounds in the periodic tables like mercury, cadmium, nickel, copper, zinc, even iron. These are the heavy metals, and when these compounds or elements are accessed in the soils, they are cause for the heavy metal stress. Okay, so phytochelatins are the peptides which have important role in the binding of these heavy metals and the detoxification of heavy metals. Okay, next, these phytochelatins are the ubiquitous in nature, means they are not only present in the plant but they are present in the microorganism and the animals. Okay, next, these phytochelatins are the short chains. Okay, thiol rich means these are rich in SH group, and repetition of peptides of low molecular weights synthesized from the sulfur rich glutathiones by the enzyme PCS, that is known as phytogelatin synthetase. So friend, this is the characteristics of the phytochelatins, means they are the short chain peptides, means generally they are 2 to 11 long peptides, but generally they are only 5 peptides, not more than this. In some cases only it reaches to the 11. Okay, then these are the thiol rich, means SH group are rich. Repetitions means these are the repetition of this low molecular weight peptides, and GSH enzyme and PCS enzymes are involved during the PCS or phytogelatin synthesis. Clear? So this is the common structure of these phytochelatins. This is the gamma glutamyl cysteanyl and glycine. Okay, so this is the structure of, we can also write as a gamma glutamyl cysteine. This is the n number of peptides and the glycine. Okay, so this is the structure of the phytochelatins. These phytochelatins have high affinity to bind heavy metals, but also they have some important role during the heat, salt, and ultraviolet and herbicide tolerance. How? Because these phytochelatins are and bond with the or connect with the antioxidant enzymes and form the synergistic defensive system for the heavy metal stress. So these phytochelatins are showing the synergistic response with the antioxidant enzymes and improve the defensive system of the plant, and also regulates the stress like that heat, salt, ultraviolet and herbicide resistance. Okay, so these phytochelatins are generally synthesized in the cytosols. Okay, and bind to the metals and form a complex, that is metal and PC complex, and transported to the vacuoles In the next slide, we will discuss the transport and degradation of these phytochelatins. So the transport is mediated by the magnesium ATP dependent carrier or the ATP binding cassette transporter present in the vacuoles. Okay. So friends, these ABC transporters have important role in the transport of large molecular weight compound in the cell. Okay. Next point is the phytochelatins, sulfahydryl SH and the COOH group can attach to the heavy metals, cation and the anions. So these two groups are attached to the heavy metals. Also there is a one compound that is known as glutathione sulfoxamine. This compound acts as an inhibitor of GSH, glutathione. Okay, and this compound is used to check the activity of phytochelatins. Okay. Next, this is the biosynthesis pathway of the phytochelatins. So here the cysteine amino acid is there, and in the presence of gamma EC synthetase, it forms the and substrate is the glutamate from the gamma glutamate cysteine. Okay, and the gene GSH1 and CAD2 are involved. In next step, glycines are attached to this peptide in the presence of GSH synthetase, and GSH2 genes are involved during this process and form the gamma glutamate cysteine and glycine residue. And final enzyme, that last step is the where the PCS enzymes is involved, and CAD1, PCS1 and PCS2 genes are involved during this reactions, and it forms the N peptides of glutamine and cysteamine, and glycine is the end amino acids. Okay, so this is a simple process where they form the phytochelatins. Okay, and Arabidopsis phytochelatins 1 and Triticum aestivum phytochelatins are the peptides or phytochelatins which are discovered in the plant. These are the first phytochelatins are discovered in the plants. Also, these phytochelatins have two substrate binding site for the GSH, which is one is metal dependent and second is the metal independent. And for the binding of this, acylation of the cytosine process is required. Okay, so friend, this is about the phytochelatins. Now come to the process of these heavy metals and the detoxification of heavy metals. Okay, so when there will be a means this is an example, means there will be a high cadmium, then there will be a binding of these phytochelatins to the cadmium, then detoxification, transport, tolerance and compartmentalization. So these processes are involved during the heavy metal detoxifications. Okay, so this is the example, and this is the structure of phytochelatins where gamma glutamine, cysteine, glutamyl cysteine and glycine. This is the phytochelatins, and this is the thiol group where, and this they bind to the cadmium and removal of two protons, and this thiol group bind to the calcium. So this is the binding process. Also you know that this is a cell membrane, cell wall, cell membrane, and this is cytosol. Okay, so these are the heavy metals which are transported through the transporters present on the cell membrane. And like there is a one example like zip transporters which have important role in the transport of zinc. Okay, so when heavy metals are transported to the cytosource, there will be an accumulation or production of the reactive oxygen species like hydroxides, peroxides, and the superoxides. So there will be a production of ROS, and this ROS directly can increase the GSS production. This is known as also antioxidant enzyme. Okay. Also, these heavy metals, since they are the phytochelatins, so we already discussed in the last slide that cysteine, glutamate, GSS from the phytochelatins, or directly heavy metals can activate the synthesis of phytochelatins. So these phytochelatins form the complex with the heavy metals, that is known as phytocelatin heavy metal complex, and this complex transported through the ABC transporters, that is ATP binding cassettes, and later on these complex move into the vacuoles where the degradation of this complex take place, and this is known as heavy metal detoxification. So friends, hope you understand that how these phytochelatins have role in the heavy metal detoxification and what is the important drug. Thank you very much